Hello! Today we will be talking about cell signaling. In detail. Now, coming to cell signaling, the very common understanding is that a receptor is there on the surface of the cell most of the time, and there's a binding component that comes and binds itself to the receptor, just like a small molecule that comes and sits on it. It's called a ligand, and after that a transduction of signal takes place, therefore giving a response from the cell. Now, coming to cell surface receptors, they are also known as transmembrane receptors. These are cell surface or membrane anchored receptors, or integral proteins to be more precise, that bind to external ligand molecules, including cytokine receptors and growth factor receptors. Now, this type of receptor spans the plasma membrane and performs signal transduction, converting an extracellular signal to an intracellular signal. Now, this can be a chemical response within the cell, or this can also be another molecule that has been activated to come and bind to the specific spot. Any of the two, it's either chemical or physical. Coming to the binding of the receptor with the ligand, this is a very beautiful situation where the ligand and the receptor bind as in a very specific spot, just like enzymes. These receptors only bind to a very specific ligand. Let's take it this way. You and your best friend saw each other for the first time and clicked so well that you decided you'll be best friends. Yeah, it's that obvious. Then, after you became best friends, suddenly your marks started either going higher or lower, depends on what you do after that. So, that is technically the signal transduction, that is the after effect, and in the end, the score or the aftermath of whatever you have done is simply the response. Very easily stated, don't do weird things, study well. Now, coming to the three types of cell surface receptors. There's ion channel linked receptor, enzyme linked receptor, and the famous G protein linked receptor. G protein is kind of like the gangster of the receptor world because it's the most important part. Now, G protein linked receptors are the ability of cells to receive and act on signals from beyond the plasma membrane is kind of fundamental to its life. It's technically like the cell has to talk to someone, otherwise it won't survive. Just like a telephone call. Now, a popular type of transmembrane receptor is the G protein coupled receptor. Many G protein coupled receptors have a large extracellular binding domain. Now, when an appropriate protein ligand comes to bind itself or to associate itself with that protein, the receptor undergoes a conformational change, and that is transmitted to its cytosolic regions. Now, the three most essential components of this transduction through the G protein receptors is the first one, a receptor with a 7 transmembrane helical segment. This transmembrane protein is hydrophobic in nature. A G protein that cycles between active and inactive forms. Now, the G protein cannot always be active because it will attract unwanted attention, and that can cause a lot of problems. And in the end, an effector enzyme or ion channel, because talking or communicating is very, very, very important. But one of the most popular responses that leads to relaxation of muscles during stress is beta-adrenergic agonists. It's a very popular type of GPCR. It is an integral protein with 7 hydrophobic helical regions. Now, beta-adrenergic agonists can also be very well known as muscle relaxants. Now, these are very important to your body because sometimes when excitement is extreme, these help maintain a nearly homeostatic state sometimes, although not all the time and not in everyone. Now, coming to a very interesting part, that is the CAMP as second messenger. In simple words, the second messenger system of cell signaling. Try imagining a relay race, and there you have a chocolate bar. You gotta pass it on to the last person, but the last person has to read the ingredients and pass it back. Imagine how painful that can be. It is kind of like that, and that is why the process takes place so fast, because they all want to eat that chocolate bar, but they cannot. So, for an example, the hormone epinephrine binds to the receptor and promotes conformational changes. It affects its interaction with an associated G protein, or GDP dissociation gets promoted and GTP comes out. This happens in a fraction of seconds. Crazy, isn't it? Love for the chocolate. Now, for all GPCRs, G protein has 3 different subunits: an alpha, a beta, and a gamma subunit, also known as the trimeric G protein. The alpha subunit exchanges GDP for GTP and transmits the signal to an effective protein. It's very much like the proteins are mobile phones finding a Wi-Fi, and the G protein is technically the Wi-Fi. Now, as it activates the effector protein, it is known as the stimulatory G protein. Now, GDP binds to its effector protein. Both the alpha and the gamma subunits have covalently attached tails. They are lipid tails. Now, the alpha subunit is the initiator of the whole thing. The GTP binds to the alpha subunit of the GS, and then it activates its activator protein. In the activated state, beta and gamma subunit also dissociate from the alpha subunit. Now, the adenyl cyclase, an integral protein, after activation catalyzes the reaction of synthesis of the second messenger CAMP from ADP. Now, the alpha subunit is bound to the GDP in the absence of a signal, of a clear signal, but the G protein is inactive at that time. The CAMP activates CAMP-dependent protein kinase called the protein kinase A, or in short, pKa. pKa catalyzes the phosphorylation of serine or threonine residues of target proteins. Inactive state of pKa has 2 identical catalytic sites, C, and 2 identical regulator units that can be called R. Now, upon binding of the CAMP, that is the allosteric activator, it binds to the R subunits and moves the autoinhibitory domain of the regulatory unit that yields a 2-phase catalytic subunit, that's C. That is, it's technically like a horseshoe magnet. Imagine a horseshoe magnet has been thrown towards you and you're just a piece of iron. How fast will you go towards that? Exact same procedure is followed by those molecules. Except here the horseshoe isn't really thrown anywhere, it's just there for some reason. Now, the ADP binding site of C subunit's position, ADP, for transferring the terminal phosphoryl groups to the OH groups of those amino acid residues of the target proteins. This is a very important step. The net effect of this cascade is simply the amplification of the hormonal signal. Just imagine the Wi-Fi got hacked that you were connecting your phone to, and now you can connect a lot more devices to it. It's technically how the region span increases. That amounts for low concentration of the hormone. That is, for example, epinephrine. This transaction has to turn off after the stimulus has ended, and it does. It's simply like you switch on the fan rotates, gives you air, and then you switch off the fan and immediately everything's gone. In a very simple way. It's not like a fan. I'm just giving an example. Now, there are three mechanisms for the termination of the cascade reaction that we just talked about. Now, coming to the word cascade, it's technically a very simple term that defines a number of reactions taking place one after the other in a very coordinated way. Now, if we want to simplify the understanding a little more, you can take a cascade as passing a chit with solution to the hardest math problem all around the class. That's technically passing of information. And that is exactly what cell signaling is about. It's passing on the information that the cell needs something or doesn't need something. Any of the two. Don't ask me, how would I know what my cell needs? They would know, ask them. The cells, I mean. Now, the response of the beta-adrenergic stimulation will end at low concentration of hormone, that is, the signal molecule. Now, it is a very important thing to remember that beta-adrenergic stimulation happens only when there is a very strong signal of the hormone. Now, the hydrolysis of the GDP-bound G subunit, that is the alpha subunit, by intrinsic GTPase activity, and the removal of CAMP, that is the conversion of CAMP to 5-AMP by a cyclic nucleotide phosphodiesterase. It just happens in a way to terminate the whole cascade reaction when nothing is needed. All of these don't happen at the exact same time, but any one of these happen. Now, in a very easy explanation, this is how the G protein coupled receptors act. As you can see, that's the plasma membrane, and that is the G protein, and how it binds and the signal transduction takes place. That was all for now, but we'll be discussing some other signal molecules that use CAMP and a little bit more in detail in our next video. So, thank you for watching the video. Like, share, and subscribe, and also study.